Of all the presentations they had at today's meeting, I thought this was actually the most important and the most interesting. And this was an update on the emerging variants and how to think about the vaccines in light of these variants. So I thought this was a really important one and found it pretty interesting. So some background, the viruses are constantly changing through mutations, so new variants are expected. After emerging, some disappears, others persist based on fitness, how well they infect people, and how well they spread. And they are studying these variants to understand whether they spread more easily, they cause milder or more severe disease. Can they be detected by the currently available diagnostic tests? Do they respond to the therapeutics that we have? And do they change the effectiveness of the vaccines? So in collaboration, they classify these into variants of interest. So these are ones they have genetic markers that may change things. Then there are variants of concern. These are variants that we believe have increased transmissibility or more severe disease or reduce effectiveness of our treatments and vaccines or diagnostic detection failures. These are the ones we were most interested in. And then variant of high concerns, this is ones that we're really, really sure are bad. And currently they have not counted any of these as variants of high consequence. These are the variants of interest. We'll skip those because not too concerned about them. Variants of concern, these are the ones we're concerned about because these are the ones that cause some problems with either treatment, detection, or transmissibility. So these are ones that probably most of us are familiar with. The B.1.1.7, that's the UK variant, the B.1.351, the South African variant, P1, which is the Brazilian variant, and then these are the ones that were found in California. These are fairly similar, and this tells you the attributes. This one is increased transmission, but the current vaccines work very well against them, and the detection works fine, and the current therapeutics work fine. This is probably the most problematic one, the South African variant. So it has increased transmissibility, but it has reduced neutralization by vaccine sera. And some of the current monoclonal antibody treatments do not work well against these. And so there are some new monoclonal antibodies that are in development right now that we talked about in this channel in the previous few videos that specifically looked at this variant and to see if it works for these, and it does. So that's good news. This P1 variant is very similar to the B.1.351 rather. And you see that their receptor binding domain mutations are very similar. This one has the same problems with immunogenicity and is not well neutralized. And these ones are a little less well known, but they have a modest decrease. So these are probably not quite as bad as these. All right, so let's look at, so in the US, it looks about 6 to 9% of SARS-CoV positive cases are sequenced. So, you know, the PCR tests that we get to determine whether people have COVID-19 or not, those are just detection tests. They don't tell you which variant you have. Then they specifically take 6 to 9% of those and they sequence the entire genome, and then they can tell which variant it is, or they may find a new variant that hasn't been sequenced before. So you can tell that they have been ramping these up. Initially, we were not sequencing any of these, and we've in the past several months have really ramped up our sequencing. The UK and other countries were doing a much better job than we were, and then we decided to start sequencing more. So these are all listed in the GIS aid. You can go to this website. So you go to GIS aid. Here it is. Yeah, so if you look at this, oh, they've changed this since I've last been here. I mean, I don't know how to read this data, but it's very interesting to look at the phylogenetic trees. It kind of makes a pretty picture, if anything else. Anyway, while that's loading, This I thought was a very interesting slide because this shows over time. So this is in January, and this is as of 5/8, so as of May 8th. So you see here, these are the original strains here, were the dominant strain in America in January. And as we go, you see that the B.1.1.7 variant has become the dominant strain. Now this one is not that worrisome, as our vaccines and therapies work just as well in these. But here is the one that we need to worry about, the B.1.351. It's currently 1%. If this is to start expanding and becoming this large proportion, I think we'll probably run into some serious problems, because this will run through the currently vaccinated population, because vaccine effectiveness to this variant is not as good. And I think there is a slide in here about that. This is showing across the nation how the variants differ. So, some areas like the Northeast have a lot of these different. Let's see, I think I've made this thing too big so you can't see the legend. Okay, here we go. So the blue lineage is the original strain. The variants of concern are this tan area and this area. So if you look at California, most of ours are the variant of concern. Same with Texas in the central area. The vast majority are variants. So as you can see, the dominant strain is this light tan, which is B.1.1.7. So that is becoming the dominant strain. The blue, which is the original variants, are diminishing. Okay, these are talking about how well the current vaccines protect us against the new mutation. And they're saying that the current vaccines seem to be okay for these, but at some point it drops off, and we don't know what the threshold at which it drops off. It gives us not enough protection is. Okay, so this is a slide I thought was pretty interesting in terms of reduced neutralization activity of vaccine sera. So they took vaccine sera after people took one of these mRNA vaccines, Pfizer, Moderna, or the AstraZeneca, or the Novavax, and they see how much worse is it against these variants than against the original strain. So if you look at this, so this is a logarithmic scale. So this is 1, 10, 100, so it's logarithmic. So this is about 2. If you look at the B.1.1.7, it's about twice. It's a one-fold reduction. So it's like 50% as good. If you look at the 351 variant, it looks like it's probably like 8 times worse. And then the P1 is maybe like 2 or 3 or 4 times worse. There's not very many studies on these other variants, so it's hard to make too much of a call on this. Maybe there's a few studies on this 429 variant, and that looks fairly similar to the P1 variant. But as you can see, it's clear that this 351 is definitely the worst. It has the most fold reduction in neutralization activity, so it's like 8 to 9 times worse. And here's the vaccine efficacy against the various variants. So the Janssen, which is the Johnson & Johnson vaccine, was like 52% effective against 351. Pfizer, they said they think it's about 75% against the 351. I don't know how big this study was. There was a study where they thought the Pfizer vaccine was like 90 or 100% effective, but it was a really small study, so you can't read the confidence intervals really wide on that one, down to like the 50%. So I don't know where the confidence interval here is, but it's definitely less effective, because against the wild type it was like 95, then 90 against B.1.1.7, and then 75 against 351. Novavax is also, you see a similar pattern. the 96% against the original strain, 86% against the B.1.1.7, and then 51% against 351. The AstraZeneca was 84% against the wild type, 75% against 1.1.7, and then only 10% against 351. So all the vaccines have a major drop off when you look at the 351 variant. Yeah, so does it cause more breakthrough cases? I think, yeah. So the conclusion, breakthrough infections are more frequent with variants of concern. So very true. What this means is essentially, in order to get herd immunity, you need to have, based on how transmissible the disease is, you need to have like 50 to 70% of the population vaccinated with 100% efficacy, um, with 100% efficacy vaccine. So no vaccine is 100% efficacious. If it's 75% efficacious, like this one, then you would need to have basically 100% of the population vaccinated to get herd immunity. If it's 52% effective, even if you got 100% vaccinated, it would still not give you herd immunity because you effectively only have 52% of the people protected. So without a booster of some sort that covers this variant, it's very unlikely that we get herd immunity. So updates to vaccines to address the variant. So they agree that periodic updates are likely needed. And the other question is like we talked about in a previous video about when does this end? Let's say that there is yet a new variant that comes out with a different immune escape mechanism, and then we're just kind of starting again, depending on how much escape that has from any boosters that we give. Now, the good thing is that when we look at the variants that we've had over the past several months, there haven't been any new variants of concern that are significantly different than the current ones. So it looks like there's some convergent mutation, that there's only a certain number of mutations that the virus can make that are beneficial to it and make it allow it to escape or make it more transmissible. So it may be that these variants we have now are the kind of the most optimized virus there is, and then if we just take care of these variants, we will that'll be the end of the road for them. So I'm hoping that's the case, but we don't know. But this was a very interesting presentation that shows that there is growth of the variants in the US population. Currently, the B.1.1 is very low, but if this expands, we are going to have problems again because our current vaccines are not really that effective against this. It's very effective against this. And so my prediction is there is most likely going to be a booster for this variant to take care of the 451 position amino acid mutation. And that is probably coming down the pipeline, and we probably do need it. And if enough people get that, and that's effective, and there's no further mutations, which if there is convergent mutation, there should not be any further mutations that are that affect vaccine effectiveness, then that will kind of be the end of the pandemic, and hopefully that's how it ends. All right, thanks for watching.